This is Jennifer Haller. She's the first person in the world to be given a potential coronavirus vaccine.
Everybody's feeling so helpless right now, and I realized that there was something that I could do to help.
First time.
Here at this research centre in Seattle, 45 volunteers are being given 2 injections 28 days apart. This is the first phase of the trial to test the vaccine's safety in a small group of volunteers.
Well done.
You know, there's obviously a certain amount of nerves associated with it, so I hope there's no crazy side effects.
The next phase will trial the vaccine in a few hundred people, then a few thousand. That will take a while, at least 18 months, to make sure the vaccine is safe and effective.
This is the first trial of this vaccine, so our goals are pretty basic. We want to look at the safety, and then secondary to that, we want to look at the immune response.
The vaccine the people in Seattle are being injected with is cutting edge. Most vaccines use a weak strain of the actual virus, but this is different. It's a genetic code that instructs their cells to make the same protein as found in the coronavirus. The body processes the protein and leaves behind protective antibodies. When the coronavirus comes along, those antibodies grab hold of the spikes on the outside of the virus and prevent it from getting into your system.
So we've got it. Fantastic.
Researchers at Melbourne's Doherty Institute were the first to grow the virus outside China, then share it with the world. The same group has just figured out how the body fights it off. Taking blood samples from one of the first Australian patients, they found the virus activates a number of immune cells in the blood to combat the illness. It mimics what happens in the body when we get the flu.
At 3 days after hospital admission, we could see emergence of specific cell populations in the blood. We knew that those populations, when they're in blood, they precede patient recovery.
And it could give doctors new insight into which patients might get sicker.
But we're eventually hoping to find some markers that we can screen the patients as soon as they get into the hospital, and that you can say up front, like, this will probably be a severe case and this will probably be a milder case.
With the illness spreading rapidly, the scientific community remains under pressure to find a solution.